huge technology because with it, you can profile anybody. This is the Gattaca type technology. So with this, with just some cells or blood, I need a source of white cells. Remember, red blood cells are no good. Does anybody know why red blood cells are useless through the DNA chip? Because no nuclei. Red blood cells, anucleated. It's just a bag of hemoglobin. Dead end. Lives for a couple of months and then it dies. No nucleus. So you need white cells. So chips, how does it work? You've got a chip, which is the surface, right? It's about so big and it's in a little plastic holder. Shove this into the machine and you read it and everything's like, how does this work? If you scale up a part of this chip, it's got a grid with little dots. And I'm not going to draw all of these, but you guys get the idea. These are fun to draw. And an average chip will have hundreds of thousands of dots. Okay? Every dot, this is called photolithography. So it's covered with a mask. And you synthesize generally a small oligo. I'm giving you guys the simplest version. There's many, many variants. So you put a grid on it, which say covers up all these cells, but it leaves a couple open. I'll put them in green. So whichever cell is open, that gets an A added for adding A. You're making oligos. You then put another mask on it. They're perfectly registered, right? Because these guys are on the order of microns across, so they're tiny. You then put another mask, and you then do C, or whatever peptide it is you want to make. And this way, you build on this grid little oligos, and you know exactly what is in each one of these holes. And just like bridge amplification, remember we went over this for next-gen sequencing, you have a localized zone containing all the same oligos. Oligos are usually about 20 meters. Got it? So if you look at this chip on its side, or on an angle, it's going to be a lawn of little oligos sticking up there. And the oligos, you know, based on a Excel grid. In fact, when you get the chip, they mail you the grid telling you what all the oligos are and what the sequences are. Remember, a 20-mer will uniquely describe any piece of DNA essentially on Earth, because 20-mer, the chances of a random occurrence is on the order of trillions. Okay, so these are generally approximately 20-mers. And the precise sequence is known by the computer. You know what genes they match. Okay? So this is awesome because now this is the technique of hybridization, which I taught you. Hybridization is taking double, most guys got this question first. You take double-stranded DNA, you convert it into 2 times single-stranded. Right? This can be done with heat or chemicals. Chemicals. The example I gave you was base, OH minus. OH minus basically blows the strands apart because you're building up all this negative charge, all the phosphates. Everybody got it? You then take this mixture, if I'm doing a DNA chip, the name before the chip is always the species that you're examining. So in a DNA chip, I'm looking at DNA. And I'm looking, maybe it's a SNP profile, so I'm uniquely identifying people. Or it could be CODIS, it could be looking at STR, remember, short keys, whatever the application is. Or it could be disease. So I'm looking for susceptibilities to certain markers, SNPs that are associated with cancer or diabetes or something like that. So this allows you very quickly, within a few hours, an afternoon, to profile this person on the order of hundreds of thousands of different genes. A lot of them are repeats, so they're designed to do accuracy, like you'll do maybe 5 or 6 oligos for different sections of the open reading frame. That's 2. So you take cells or white cells or scrape the cells from cheap. You bust the DNA open, you get out DNA, you put it on this chip, you denature it, you let it rehybridize. You first treat all the DNA, so this is patient's DNA. You put a FLD, is short for fluorescence. So put a fluorescent tag on one end. You can do this chemically easily. Remember, we did the same thing with next-gen sequencing. Usually put like a barcode or some kind of, something that allows you to determine what it is. It's usually a fluorescent one. So you denature all this stuff. This has FLD markers. These rehybridize, right? These different spots on the chip. You then shine a light at it and you get the grid pattern. And the grid pattern will be each one of these little dots, just like next-gen sequencing. We're looking at the fluorescent light. But here, the light, the intensity of light is proportional to the amount of DNA that hybridizes. So if the patient has not much DNA, you'll get a small signal in a second. So the intensity of the signal will be a function, say, of the number of copies of a particular gene the patient has. Got it? Does that make sense? That's how it should work. RNA chip, very powerful because you can look at expression. You can look at expression of some deleterious gene, like BRCA. Abelson tyrosine kinase is associated with cancer. So many cancers have this gene. So if you have maybe 3 or 4 of these dots representing that fusion protein, which is oncogenic, causes cancer, you can get a signal. RNA, it also gives you intensity. So if you see a lot of fluorescence, it means the patient has a lot of this type of RNA. There are also protein chips, which I'm not going to go into. But this is how all the chips work. So it's a combination of oligos. There are other types, but just to work with them. Oligos is the simplest type. So it's oligos, the exact sequence is known by photolithography and computer. You sell these and Affymetrix sells hundreds of these for all different kinds of applications, different diseases, different types of profiling stuff. Okay? RNA, DNA, protein. Where are we? Any questions on chips? Any questions on chips? So it's oligos, hybridization, right, denaturation, hybridization, and then reading like a fluorescent read, just like next-gen sequencing. So basic steps in bioprocessing, we went over this before. Now the next bunch of slides I'm going to show you is the entire 195 course. This is the nutshell. So 195 is all about this process. So huge amounts of cells are obtained from a bioreactor, say 20,000 liters. These are kilos of cells. The cell paste is revolting. It resembles human excrement, and it fills many garbage, many bins this big. Frequently what is done is either the cell lysate or cell-free media is often concentrated, frozen down, and shipped out because it's too expensive to work it up in this country. So a lot of this is done, for example, in the Caribbean. And the cells are busted, or protein product is secreted, which is the favorite way, extraction of free protein preparation. Concentrated, this is often frozen and shipped out, as I said, to some place where labor is cheaper than in this country. Chromatography, and I'll talk about chromatography quite a bit. Again, this is a focus of all of Dr. Doreen's lectures in the spring. So she and I co-teach this class, and in spring you'll hear Doreen, and she talks about downstream purification. Upstream is anything with live cells in it. Once the cells are busted or you've removed secreted product, it now becomes downstream. So concentration, this is ultrafiltration, and in the spring we have the expert, John Rosenberstein, who will be lecturing again this year on chromatography, stabilized final product, drying, usually, remember these are protein products, they usually have to be lyophilized or freeze-dried, so you end up with a powder in a vial. The final product is packaged, labeled, and then out it goes, and it goes to customers. Usually quarantined for a period of time. So this period is upstream. This process is upstream. This is downstream. Downstream purification. I went over this last lecture, and it was over quick. So upstream, selecting cell to be used. Again, this is that little vial that I erased over there, which is stored under lock and key. This represents the company's ability to make that product. Fermentation process is well understood. Bacterial, large quantities, easy to alter genetically. Increased level of production of bacterial protein by introducing additional copies of relevant genes in the host cell by recombinant DNA technology. It's a huge amount of engineering going on here, which I'm glossing through in this class. It's pretty much an introductory class. But there are many ways of manipulating the bacterial chromosome, or the host chromosome, so that it becomes better able to produce huge amounts of recombinant protein. Now, bear in mind, as I've said in earlier lectures, anytime you're forcing a bacteria by using usually super hot promoters and operating system, so if you're forcing them to make insulin, which is completely worthless to the bacteria, and they're diverting a huge amount of their energy in order to make some human protein, obviously this is building up and it's toxic to the bacteria, and it's sort of a lethal blow, right? So if you divert an organism to chugging out tons of protein that you want and they don't want, obviously bacteria are suffering massively. This is usually a process that we trigger when the cells are high up in the exponential portion of the growth curve. So you have lots and lots of very healthy cells before you trigger them to start producing. Most common bacterial species used today is E. coli, and sometimes in diseases are using others. E. coli is what's common. Selecting cells are used as a protein source. Bacteria make the majority of protein synthesized naturally by E. coli are intracellular. This is a huge problem. Let me explain what it is. This is a problem with E. coli. This E. coli doesn't secrete much stuff. It can secrete certain types of hydrolases and digestive enzymes. It's difficult to get it to do that. So here's a bacterial cell, say E. coli. No nucleus, and it is producing tons of recombinant protein. But how do you get it out of there? Okay, you have to crack these and bust these cells open. There are many ways to do that. So you break it open so that your protein now comes out as accessible for you to try and purify it. The problem is when you bust the bacteria open, all of the thousands of proteins that are in this bacteria are spewed out into the media along with what you want. So downstream purification, i.e. trying to get insulin, a growth factor, or an antibody out of this mix is tough to do. That's one of the problems with E. coli. Downstream purification is a much bigger challenge. So you've got to break them open and release all this garbage into the media. Then you have to get your protein out of it. Okay? If you pick a CHO cell, which is massive, so here's a CHO, you can engineer it so that the protein you want, so the transgenic recombinant protein, is secreted. Okay, remember in this class and in bio, we've talked about the endomembrane system. So you take advantage of dynein and kinesin. Remember, they're going along the superhighway and they're dragging those massive organelles packed with protein, and then they're secreting. Now the beauty of this is now your protein is dumped outside the cell. So you don't have to bust this guy open. So almost all recombinant proteins made today are made from mammalian cells, just because of this problem. Plus that, this is a prokaryote, and this is a eukaryote. Eukaryotic cells make proteins differently. They have different glycosylation patterns. As everybody knows, maturation of the protein through the endomembrane system is unique. These guys do not have it. So this is a big plus for using CHOs or some other group. CHOs probably most common. How hard did I know? Genetic engineering by fusion. Okay, I'm talking about fusion. This comes up on the next class. Okay, here's some protein you want. Let's say it's growth factor. So here's growth factor. And it's being made in some organism. Let's say you're making this in people. And this is growth factor. Human growth factor. A trick that makes it much easier to purify this is something called a fusion protein. So you fuse this guy in frame with another protein. This together is called a fusion. This is something that's easy to grab hold of. So this protein's easy to purify. This one's not so easy. You can purify them because they're covalently stuck together. There's a peptide bond connected. You then purify this by grabbing the green one. You split them apart again, which is trivial. I'll show you in a second how it's done. And now you get your protein out essentially pure. Very powerful. In addition, fusions are usually more stable. So it helps stabilize your growth factor and it helps you purify. So these are good ways, good technique to do in E. coli that allows you to get your protein out of this huge mess that I just described. So genetically, how do you do it? Dead easy. If growth factor is here, and I'm skipping all the introns now, so here's growth factor. So here's a stop. Here's your AUG start. You chop all the stuff away. Usually there's an MCS, multiple cloning sequence. It's also called a polylinker. Everybody remember this? Multiple cloning sites. Poly. So you just cut in here. It's got to be in frame. And you fuse some protein that's easy to grab. I believe in the example I used, so here's the new AUG, and the promoters and everything are all up here. Remember, Tata, cat box, all the -10, -30 stuff is here. Now, when the ribosome comes on, this is made to mRNA, ribosome comes along, and he chugs right through this whole thing, because this stop you remove. So this is all in sequence and in frame. So you can go right through the C-terminus of your catch protein. I think it's called MAL-E for maltose binding protein, I think, in the next slide. I think it's MAL-E. But there's many things. There's GST, there's all kinds of stuff. MVP, maltose binding protein. This is polyculture MVP. The stop is removed, and you put it in frame so that you have your protein here. Everybody got it? This thing is made as a single polypeptide, which you purify, and there's usually a cleavage site here, protease. So you want to clip this protein and get your protein out. Everybody conscious? Has this gone too fast? I don't see anybody really sobbing out there. Okay, try this picture. Please be sure you understand all these elements here. It's typical plasmid map. Can you see it? Typical plasmid map. MAL-E, maltose binding protein. This codes for, is it saying it over here? Secreted protein will carry target gene outside cell. Fusion protein, maltose binding for affinity chromatography. TAC is just some powerful promoter. Here's MAL-E. Here's a spacer. Come on, don't panic. Work with factor 10A. Factor 10A is in the coagulation cascade. And when you guys take the drug class, 405, in the spring in a couple of years, one of the teams will present this. So factor 10A. Factor 10A is coagulation. It's involved in prothrombin and cleavage of fibrin to make clots. Okay, so factor 10A is a unique sequence that it cuts. The factor 10A site. Target gene in purple, yay, I guessed right, just like I showed on the board. Lac Z, more on that in just a second, and a strong terminator. Okay, BLA is beta lactamase. This gives you resistance to the penicillin family, such as AMP. Okay, this, BLA, what do we need BLA for? Anybody out there? Why do you need BLA? Why do you need BLA? You must have this in the plasmid. Why? Anybody besides Matt? Tell me.
You then put the same resistance tube in there and it kills off the cells with them.
Any cell that has picked up this promoter, this plasmid, will survive if you plate it on penicillin. So all you do is have agar, you put amp in there, you put all these bacteria. The millions that do not have this plasmid die, and the few that don't survive. You pick the colonies out, they will have this plasmid in it. The colony is growing cells, and they will hopefully have remology in the egg. Okay, this vector is called PMOL, and you can buy it. It's called PMOL because it gives you this binding protein, and it's fused in frame. There's probably a poly here, polylinker shown right here. Polylinker allows you to cut. Many of the sites are in frame, so we'll leave it in frame so that this and this are in frame. Okay, moly is at the end terms, max.
What is the spacer?
Spacer is, if you think about it from a 3D perspective. If this is too close together, factor 10A, which is a fairly big molecule, has trouble getting in there and cutting. So usually just put a linker. The asparagine acid is asparagine. This is asparagine. It's one of the 24 acids. And there are 10 of them. So it makes a long, random coil piece. It just opens it up so that there's a space between these two. It allows factor 10A, which is a protease, to get in there and hit that site. Cut this away. Okay, so very quick, I have to explain about chromatography. Everybody understand this concept? Our fusion protein, and it's replicated in this plasma. Okay, what's chromatography? Probably 95+% of downstream purification is called column chromatography. The reason is it has a large column. So the column is often a glass tube like this. And you have an inlet, often the inlet on the bottom. This is inlet. And out here is outlet. Okay, columns have packing, and they have frits. So there's some sort of filter here. And this thing is filled with packing material. Here's the little beads. I can draw these, but there's millions of little beads. Okay? Inlet, you pump on something you're trying to purify. For example, either busted open E. coli cells expressing this, plus tons of other bacterial proteins, and you want to get this guy purified. So you pump this guy on. There's some cleanup steps you have to do. Centrifugation. You pump this on. The beads are stationary. This is called the stationary phase. So all chromatography and even extractions. No, I don't think you guys have done liquid extractions in lab. That comes in order. So next semester, you do liquid extractions in the Orgo lab with Dr. Rose. Similar concept. There is a stationary phase, so the bead represents the stationary phase. All chromatography is based on this concept. If you understand this once, you'll have to get it again. The purple liquid flowing through here is the mobile phase, the mobile. As you pump this on, protein, junk that has affinity for the bead will spend more time stuck to the bead. Things that have no affinity or binding capability will flow right through. As a result, you end up with what's called an elution profile. Okay, this is the basis of all column chromatography. This is easily 95% of downstream purification. This process. And there are literally hundreds of different packings that you can buy. The most common one is ion exchange, where you have a fixed positive charge. So the stuff that's plus or neutral will flow through, and stuff that is negative will bind. This allows you to partition or to enrich the outflow or the eluent with stuff that your protein has that property. Okay? That's how this process works. In the case that I've given you, you have the bead in green. This has maltose. It's made of a maltose polymer. Maltose, for those of you that did bio or had bio, what's maltose? Glucose disaccharide. So it's two glucoses stuck together, and it's a key element in starch. So almost all biological polymers will have maltose in it. So you can make this bead from any kind of starch, for example. So there's cross-linked starch and cellulose and all that stuff. So as long as you have glucose in here, two glucoses together, in this bead, this protein, mol binding protein MBP, which is coded by mol E gene, will bind to it. And it will have growth factor, whatever you want, stuck to it too, because these guys are connected. Everybody follow? So you wash this column, you get all the bacterial garbage, enzymes, ribosomes, all that stuff goes through this thing, and you end up with this fusion protein stuck. What many people do is they then load the column with factor 10A. Cuts all these bonds. You then wash it, and in the eluent comes out your growth factor free and clear. That's factor 10A, but that's easy to get rid of. Instead of having thousands of bacterial garbage proteins, you end up with factor 10A. That's a typical way to do this. Okay. The other way is to elute the whole thing and then split it into column chromatography basis of 95% of downstream purification is some kind of packaging. It's all stationary versus mobile phase, and how much time each of the components of that gamish is sticking to. Okay, everybody understand?
The mobile phase is just some sort of buffer.
It's buffer. It's usually buffer. And there's usually some kind of eluent. So you usually add something that will bump the product off. In this case, it's maltose or glucose. So you just pump glucose onto the column. And everybody that's stuck now, instead of binding to the matrix, now binds to free-swimming maltose, and it all comes off. Bump off. 10-minute break. We reconvene at 10 o'clock. Completely differently than we do. So eukaryotic cells are unique. This is one reason a lot of expression is done in yeast. Yeast are fungi. It is a eukaryotic cell, massive, not a tiny little bacteria at all. Yeast are similar. They also have secretion processes. So yeast is a good second candidate if E. coli is not working right. Okay? So prokaryotes are cheap and easy, but they have many downsides. So advantages and disadvantages of producing proteins in bacteria. E. coli genetics are all understood. Almost unlimited quantities of protein can be generated. It doesn't even say it. Dirt, cheap. E. coli, cheap. Right? It's an intestinal bacteria. It grows on anything. So it's cheap. Fermentation technology, well understood. So these are all pluses. Downsides. Foreign proteins produced as inclusion bodies must be refolded. This is a horrible problem. I'll explain it. Proteins cannot be folded in ways needed for many proteins to be active. Some proteins are inactive in humans. Okay, what's more? Okay, so here's our E. coli cell again. This is our E. coli cell. Okay, and you tricked it into producing tons and tons of growth factor, which I'm showing in purple. So is the mRNA, remember you've got a really powerful promoter on there, and a good operator, so there's huge expression, tons of mRNA going out, and lots of ribosomes are spending all their time making this growth factor. So growth factor comes spewing out, and as more and more and more of this builds up, it starts to precipitate and collect into massive structures called, I'm not going to put a circle around it because you guys will think it's membrane bound. These are called inclusion bodies. And they're visible on a light microscope. When you look at a bacterial cell, you see that it's packed with these structures. That's your protein, totally denatured. So it's denatured, it's all random coil, and it's in a precipitated form, roughly resembling egg whites. So coagulated protein, okay? To get your protein growth factor out of there and to get it to fold correctly is an enormous job. Whole careers of my colleagues have been spent doing refolding, and this is an art. Now, sometimes you can do it and you can get your protein out of it, but if not, then you're doomed. That's called inclusion body. It's an intractable form of your recombinant protein. The E. coli is basically trying to get rid of this stuff just by packing it into this insoluble glob. It has no way to ship it out. Okay? That's an inclusion body. Protein expression. Genetically engineered bacteria can be grown in huge scale, either anaerobic or aerobic. Right? If you deprive them of oxygen, then they're fermenting all the time. Computers monitor air to keep oxygen levels stable while the ideal phase of growth is highest. Bacterial promoter must be activated to stimulate foreign gene expression. This should be obvious, or I could just explain this briefly. So here's a growth curve, and you guys will be doing this next week with the CHO cells. So you're going to actually look at growth. So this is growth. And for any organism, bacteria usually looks like this. This is log of cell number. This is 0. And then this would be, 10, 100, 1,000, 10,000, 100,000, 10 to the 6. And some small. All cells start off at some low level, and then they have an exponential phase, and then they have a stationary phase. This is in a fermenter, for example, or even a flask. It'll look like this. This period is the lag. All organisms have a lag, including the CHOs. They've been frozen for a year. The little seed stalks that you guys will be following today in lab were laid down by the students before you a year ago. The cells were frozen for a year. They're not very happy. Many of them are dead. It just takes them a long time before they recover and they start getting up and replicating again. Now, this is an exponential growth phase. This period here is exponential growth. Promoters are designed so that they are quiet, and you can turn them on by adding something into the medium, which we'll talk about later. Somewhere of the year, you trigger or activate promoter. Transgenic promoter. Somewhere this way. You want the cells still happy and growing logarithmic, and you want a lot of them. Because every cell is going to make so much protein. If you go into the death phase here, stationary phase, where nobody's happy anymore, they won't produce much. Now, as soon as you activate the promoter, the cell stops dividing, because why? Because you've hijacked all of its ability to make anything into making growth factor, whatever you want. When you hijack the cell, it can't do anything on its own, right? It can't replicate. So all its energy is going into making this garbage protein that you have put stuff in it. Okay? It usually activates somewhere up here. The highest activate gene recombinant organisms after organisms completely synthesize important natural proteins needed for metabolism. That just says you want the thing growing, you want it dividing, you want it in this late exponential phase. Everybody understand this concept? Typical bioreactor. We have 4 of them down in the biomanufacturing lab, and you guys will get to use this semester. So this next few labs we have is basically practice for biomanufacturing when you'll be running DEX. DEX are hybridomas. So in the spring semester, you guys will be running, making antibodies. They all look about the same. It's got an impeller or motor which drives the paddle to agitate it. You usually have a sparger, which you can sparge air or O2 into. So you have ports all over this thing, and we'll go through all this in another few weeks when we start doing sales. Sparger is a way of making micro bubbles of air to oxygenate this thing so you get mass. Typical lab scale. This can be a few liters. Ours is 1 liter. 1 liter is about halfway, 2/3 up. This is typical bioreactor. Ports. All of these things are probes that are checking pH, dissolved oxygen, etc. You can monitor all this stuff real time, even growth. You can monitor. Okay, that's what all this stuff. You have a PC there that monitors the. So this is a relatively small scale. And remember, as I said before, you start with a little 1 ml vial. You guys will do this afternoon. You start with a 1 ml vial, and you end up in this, and then finally you end up in this. This is a typical large-scale fermenter or a bioreactor, where it's many stories high, and this would have probably 40, 50,000 liters. So here you're producing kilos of product. Okay, and guys running this all have hard hats on, and they're following SOPs, and they're working on the floor. Fungi, yeast is the most common. Source of wide-ranging proteins using product from the animal feed. Here, many species of fungi use as host for engineered proteins. Again, yeast. Fungi are eukaryotic. That means they have everything we do. They have endomembrane system. They have protein maturation, glycosylation, secretion, everything, just like us. Okay, from the post-translational modification, a label properly folded. So human interferons are used, for example, viral infections, such as HCV, hepatitis C virus. Patients are treated with interferons to activate the system. Lactoferrin, bovine chymosin, which is used to make cheese. Aspartate proteinase, triglyceride lipase, etc. These are all growing different types of fungi. A is aspergillus. So aspergillus is a common mold. Aspergillus niger is a common red mold, black red mold. You guys are probably, unfortunately, more than one. Protein expression, plants. So a lot of agricultural stuff is made in plants. You can also make recombinant human drugs in plants and animals. And the beauty of this is that instead of having to ship out vials of product that need to be refrigerated, you can ship out a plant or an animal, right? And then the country can grow this up and treat their populace. Plants are genetically engineered to produce specific proteins such as therapeutics. Plus encourages rapid growth and reproductive rates of plants. Tobacco plant, the best choice for biotech protein production. Genetic material is integrated. Millions of plant protein factories can fill the fields. Disadvantage using plants. Not all proteins are expressed. Cell walls, so there's some challenge. But plants is very tough. Mammalian cell culture systems are challenging. Nutritional requirements complex. Why is this, you ask? Many cells, such as our cells and mammalian cells, have nutritional requirements. Vitamins, growth factors, essential amino acids, essential fats. We are dependent on eating. So we need a huge amount of nutrient going in order to survive. Okay, so nutritional requirements can be way more complicated. E. coli will grow on almost nothing. Mammalian cells grow slowly. Typical replication time is 1 day, 1 replication per day. Compare this to bacteria, which E. coli replicates every 20 minutes. So in 1 day, it will go through dozens of replications. This turns 1 bacteria into millions. Very, very fast. Contamination yeast, the best choice of proteins, nested yeast for humans. Obviously, a very rigorous control system so that you keep things stable. Antibodies, I went over this in last week's lecture. Important molecules, plasma cells, or B cells in response to infection. Enormous therapeutic potential. Roughly half of new biologics and existing biologics on the market are engineered antibodies. So an antibody can be engineered to detect anything, as I stated last week. Remember the ELISA lab, we have an antibody that recognized chicken globulin. Light chain, variable antigen binding. You can bind anything that you design. These can be made very easy. They can be expressed, for example, in CHOs. Also hybridomas. You guys do this in the spring in 195. Biomanufacturing capacity maintenance. We scale it up a little while. Looks like this. Again, this is a ribbon diagram showing sheets and helices. And it's got the variable light, heavy domains, and then the constant. Mammals all make these. Protein expression, animal bioreactive systems. All these types of animals have been made transgenic, have been made transgenic, and they can all produce proteins of interest. Not shown are goats. Goats is another classic because goats produce milk. And you can have a therapeutic protein that's secreted into milk. So it's produced in the milk glands. So you have a specific milk promoter on there. And then all you do is harvest milk out of the goat and you feed it to a population that requires the therapeutic. And it's much cheaper to ship goats into Africa than it is to provide refrigeration and storage to treat populations. Well, often it's much better to send a transgenic goat than it is to send vials. Monoclonal antibody production, minus injected antigen, mouse secretes desired antibody purified. I mentioned in last week's lecture, you do tail snips, little drops of blood come out, and you collect it, and in there is what are called polyclonal antibodies. Antibodies against whatever you inject into them. Obviously, why don't we have human antibodies produced? Can you inject humans and have them make antibodies? Why not? It's unethical. Remember, anytime you inject some trigger with an antigen, you're causing an inflammatory response. Otherwise, you don't get squat. So you have to make the animal sick so that the immune system gets activated. You cannot do this on that. However, there are ways around it. So you can produce humanized antibodies. We can talk about that in the drug class. We'll take the class. Antibodies produced in response or reaction to antigens, invading virus, bacteria, etc. Antibodies combined, neutralize the antigen, protecting the organism. Production of antibodies part of the immune response, helps living things resist disease. This is called adaptive immunity. So it's one huge arm of the immune system we have. It's all engineered by white cells, white blood cells, WBC, specifically T cells and B cells. It's all part of the any questions? Insect system. I know it sounds weird, but this system has been around for decades. We did this when I was in grad school. All the stuff was available for almost 10 years. Baculoviruses, or viruses that infect insects, not to be confused with bacteriophage. Remember, bacteriophage or phage are viruses that infect bacteria. Baculo infect insect cells. Insect cells are not mammalian, but they're animal. An insect is an animal, and it's a eukaryote. Vehicles to insert mammalian DNA cause desired proteins to produce in insect cells, also grown just like CHOs in the aseptic environment. Sometimes post-translational modification is slightly different. The insect is not the same as a mammal. Currently used when small quantities of proteins are used to insert. A very, very good, cheap system. We use this at the company all the time. Very quick, useful method of research. Proteins expressed in poorly, bacteria or yeast are misfolded. This means often inclusion bodies, which is just not functional. Express large active organisms. And usually, if everything fails, you try insecticide. Okay, this is one of my favorite slides, and it basically compares all the different systems we've just gone over. So, bacteria, yeast. Remember, yeast is fungi. This is a eukaryote. Fungi, very similar to us. Many genes are similar. Endomembrane system, glycosylation, post-translational modification, secretion, all good. Insect cells, Baculovirus is the suitcase that you use for infecting the cells and getting your DNA. Mammalian, this is like CHOs. CHOs or hybridomas. Plants, I mentioned transgenic plants. We talk about more of this in the agrobiotech chapter, which I hope I get to in the next week or two. Little bunny rabbits, mammals. Many types of transgenic animals. Everybody got it? Speed and bacteria win every time, right? A doubling time of 20 minutes. You can go from a little vial to a huge fermenter in just a few weeks. With mammalian cells, it takes months because they divide so slowly. Yeast, single cell, also very fast, also fermented. Remember, you can go without oxygen. Sometimes that's desired. Insect cells, these guys are eukaryotes all the way down. Insect cells, a little better mammalian cells. Transgenic animals and plants, this is usually a very slow system. Cost, and the winner is E. coli. Remember, you don't need anything. You need a little sugar and that's it. Mammalian cells, brutally expensive. Growth factors, vitamins, they get sick all the time. They start slowing up, nobody knows why. Much trickier. Very easy. Yeast, insect cells, generally easier. Plants, easier. Mammalian cells, very hard. Cost is high. Glycosylation. And the winner is mammalian cells and animals, mammals. Insect cells next, plants, yeast, bacteria. Folding, and the loser is bacteria. Inclusion bodies. No means of getting rid of the garbage that you're making it make. You can't get rid of it, so it just builds up. So folding is terrible. It's very hard to get active protein out. Everybody understand what I mean by folding? Right? Proteins have a specific three-dimensional structure. And then as they come off of the ribosome, they start folding. Well, if they're coming off by the ton, they don't fold well. They start to improve. Government regs. Single cells, who cares? Insect cells, fair. Plants, animals, much tougher. So if you want to make a transgenic animal, there's higher hurdles that you have to jump through. It's a good review slide to look at all the different methods of comparing. Downstream. So downstream means you busted cells open or the product is secreted, either one. So downstream means you no longer have cells and you're now trying to get recombinant protein purified, stabilized, and bottled and sent out to the market, to the doctor's office. Protein purification, I mentioned columns. Preparing extract for purification, protein must be harvested. Usually you need some means of getting the recombinant protein away from the cell that made it. The entire cell is harvested, the protein is intracellular. This means the protein did not get secreted. Again, bacteria leak to mine. And they're very poor, if any, secretion system. Requires cell lysis, you gotta bust them open, freeze fall, detergent, mechanical methods, something called the French press, which I used to use in a lab, but I've never seen it anymore. There's a bead bottle breaker and all kinds of methods where you blast the tar out of bacteria cells to bust them open. French press, you pump this thing with your hand. It's a huge steel encased bomb. And you pump the pressure up, and then you blast it out of a tiny steel orifice. So it flies out of there, and everybody gets broken into pieces. These are different ways of busting bacteria open. Why are bacteria so hard to break open? Cell walls. They're tough, they're small. You have to do something heroic. After cells are ruptured, alcohols such as ethanol, etc., salts, are added to the mix. Agents increase interactions with protein molecules, get them out of there. Culture medium is collected, the protein is extracellular. Extracellular implies some sort of secretion system, secretory system. Okay? So you have engineered yeast or etc. in order to the protein is not secreted, it's out in the medium, so you do not have to bust them. Protein purification, step 2, stabilize. Maintain low temp, often on ice or cold rooms, proper pH. Add protease inhibitors. There's lots of proteases in the mixture, especially if you had to bust cells open. Proteases, nucleases, etc. The proteases will grind down your product and reduce your yield and your profit margin. Antimicrobials to prevent proteins from being digested. Important to remove these additives later. Add additives to prevent foaming. There's anti-foaming compounds. These are certain types of surfactants, because foam, as you blow air and paddle stuff up, foam rises, and the foam can rise right up through the whole system, leading to contamination. Additives, everything obviously all has to be removed. You can't shoot human beings up with some soap that you use to keep reduced foam. Separating components and extracts. Similarities between proteins allow them to be separated from other natural molecules, starches like better versus proteins. Differences between individual proteins are used to separate target proteins from others. This is usually polychromatography. There's some for that's this stuff. Protein precipitation, very common. Proteins quite often have hydrophilic amino acids on the surface. Characters used basically for separation. Salts, such as ammonium sulfate, which you guys did make this semester, but we've done in past years, precipitate protein solvents such as ethanol, isopropanol, acetone, ethyl ether, etc., can cause precipitation. So make your protein fall out of solution. Hopefully in a form that can be full and can be maintained. Centrifugation separates samples by spinning. This is often used to get your protein free of cellular crud, broken cells, etc. Proteins are isolated in a single layer or separated from heavier cell content. Showing typical types of centrifugation. We have this type in lab, and you guys will do this today in lab. You'll separate and spin out mammalian cells, CHOs. You'll resuspend them and then spin them out to get rid of all the stuff that's in there. Before centrifugation, you pellet cells at fairly low speeds. It's very cheap and easy to do. And this is a rotary, continuous type of centrifuge, which allows you to purify your proteins and separate it from cells real-time constantly. So you can have these massive fermenters fed right into this thing that spins all the time, and out comes solid protein, solid cell debris, and soluble material that you move on. These should run constantly. Separating components extract. Size-based filtration method, membrane filtration. Again, John Rosenbergsky will lecture on this in the spring. Thin membranes of nylon are engineered that vary in pore sizes. Faster method of centrifugation, microfiltration, ultrafiltration. We'll go over all this in the spring. Microfiltration is getting rid of precipitates and bacteria. Ultrafiltration is usually to separate proteins and nucleic acids and make better. Some ultrafiltration processes can separate large proteins from small. Membrane filtration, obviously, which is probably the slide. Size-based filtration, diafiltration and dialysis. These are both methods to separate. Dialysis basically separates your protein from small ions, such as salts, glucose, etc. Metabolites can be removed. Dialysis used to remove smaller salts, solvents, and other additives. Salts are replaced with buffering agents to stabilize proteins prior to purification. This is some dialysis system. This would be like a dialysis bag. And again, those of you in biology, we had a dialysis and osmosis lab, and you guys did exactly this. So you take a bag with a high salt protein solution, seal it with clamps, chuck it in some low salt buffer, and the salt all equilibrates, then you end up with less salt. This process is called dialysis. Same thing is done on kidney machines. So patients that are diabetic or patients that have kidney problems are put on a dialysis machine. It's the exact same thing. So you take the patient's blood, you pump it through a series of dialysis bags, And you get rid of urea and all the kinds of nitrogenous that the patient can't get rid of themselves. This is like a typical lab column, and again, you guys will do this in the spring. We have multiple variants. In 195 class, small column is used, and this is a massive column. This thing is probably 10 feet or so in diameter. The human, I don't see humans here, but you can see this floor space. So this thing is probably about the height of this roof. It's probably about 10 feet diameter. Same thing. It's a column. It has a stationary phase, and you pump liquid through it, and stuff binds, and this allows you to purify your protein away from other stuff. Separating components and extracted methods and protein separation. Chromatography to sort proteins by size or binding affinity, etc. Long glass tubes, such as this one, filled with resin and buffer, and the resin beads, which is this white gel you see, has fixed charge, maltose, glucose if you're using maltose binding protein, etc. So you design a stationary phase so it can interact with the desired protein that you're trying to purify. It allows you to wash everything else out, or most everything out, and then elute the protein with it. Long glass tubes filled with beads, protein extractors added, flows through. Depending upon the resin used, proteins either sticks to the beads through the glass column, or is filtration, which is called gel filtration. Protein purification, separating components and extract. Size, exclusion, chromatography, called SEC. This is one of the workhorses of biochemistry and chemistry. I'll explain this. Gel beads is a filtration system. Large protein molecules work their way around the beads. Small proteins go through. Gels available in many pore sizes, and correct gel separation based on molecular weight proteins that requires large volumes and make good for preliminary separations. So let me explain. Okay, they're showing the column. You usually run these upside down, but I'll do it the way they're doing it. So here's the column. This is the inlet. Column. Again, there's some bead in here, like packed gel back. And this contains beads. So it has a bead, and the bead has some sort of cross-link on it. This is called size exclusion chromatography, or SEC. And again, Dr. Osgood will go over this in detail and explain. Have the bead. Put proteins on it. Big stuff that can't penetrate the bead. The orange is large. Large proteins can't penetrate the bead, and they flow around the beads and they come out. Medium or smaller size stuff penetrates the bead some percentage of the time. So medium or small gets stuck in the bead, and it takes a long time before it comes out the bottom. So if you monitor protein elution, or proteins coming off, remember this is the exact same as the system I just described. The stationary phase is the volume inside the bead, because anybody that goes in there is moving slower than somebody that doesn't. As a result, look at protein concentration. This is UV light, optical density, etc. And this is usually called column fraction. So you bop along here, you see nothing. All of a sudden you see some large peak come up. This guy is large. Big proteins come out first because they are not spending time in the so-called stationary volume. Little stuff comes out at the end. So we were showing the line thing. So here comes stuff in the middle. And then at the end comes stuff that's small. And I have this small. So this is kind of counterintuitive. Big stuff comes out first. You guys are used to polyacrylate agarose gel, where you set up a massive electric field, whenever you plug it in, you've got minus and plus, then you're ramming everybody through a gel, which is a matrix, and as they're fighting their way through, they're taking either a long time, meaning they're really big, or they're small and they zip through. This is different because the bead has a volume inside of it. So big stuff comes out first, not last. A little opposite of what you're used to. Simply because the little stuff is spending a lot more time inside of the beads. So this picture is showing you big stuff, which is ripping through the interstices, and little stuff is going slowly, just getting trapped inside the bead volume. So if you think of this as 100% of the volume is this whole thing, as much as 70% of the volume can be inside the bead. So as a result, we get a big separation. Got it? Size exclusion. Mixed up exclusion. Ion exchange relies on electrostatic charge to bind proteins to beads. So ion exchange will look just like this, except instead of size exclusion. You now have, say, fixed charge. So in black is called ion exchange chromatography. It's usually called cation exchange chromatography or anion exchange chromatography, depending upon what you have. Now this packing, for example, if it has tons of positive charges on the beads, then a protein that goes through that's mostly negative will go slower because it's going to be bound. Positive, negative, trap. This whole process is called ion exchange chromatography. It's either exchanging cations or exchanging anions. So in the example I gave, this, with fixed pluses, is anion exchange. Because negative stuff is binding. So this is showing fixed pluses, you flow stuff through, bunches of proteins come out, and usually in order to get them to a loop, because some it's got a lot of negative charge, the protein can be stuck really tightly, you increase the concentration of sodium chloride. Sodium chloride is Na plus Cl minus. I think Dr. Bose has lectured on this. Metals on the left tend to become protonated as ions like Na plus. And halogens on the right tend to pick up a negative charge to achieve an octet, right? Ionization. So lots of sodium and chloride will bump stuff off that's bound. Because now you're breaking the ionic interaction by causing anions, in this case, to spend more time binding to sodium than with this fixed plus charge ion. Affinity. The third type of drug, the SEC ion exchange method, relies on the ability of most proteins to bind specifically and reversibly to some kind of ligand. Small molecules that bind a particular large molecule protein, such as a lock-in-H system. The classic example, maltose binding protein, which I just gave you. That's an example of affinity chromatography. You're relying on protein to have an affinity for sugars. After proteins are bound to resin, buffer solutions wash out. Special buffers, such as containing glucose, are used to break this bond and stuff gets released. It's quite often used for purification of fusion proteins. Mol-E. I just spent about half an hour on it, so hopefully everybody has a grasp. This is attempting to show a lock and key, where this is some affinity ligand, and the whole mixture goes through, and if this is maltose, and this is maltose binding protein, the MAL-E gene product that I just went over, this will slow down, and that stuff will bore through. Okay? Well, a question of partitioning. I have molecules between the phase not moving and the phase moving, mobile and stationary phase. Okay? HIC, this is the fourth that you're learning. Hydrophobic interaction. Same thing, proteins are sorted on the basis of hydrophobicity. Proteins are coated with some hydrophobic molecules. These are usually hydrocarbon chains. They're very cheap, very fast, and very good. Hydrophobic amino acids are attracted. You will lose with decreased salt and protein appears. So decreasing non-polar character of the buffer enables hydrophobic portions of each to bind to the resin. Proteins are eluted by increasing non-polar character of the buffer, i.e. getting rid of salt. So you usually run a negative salt gradient. The blue line is showing you conductivity, which is electrolytes, sodium chloride. This goes down, so you wash the sodium chloride out. And there's simple ways of programming gradients like this. That's trivial. IEF. It's the fifth time you're learning. Isoelectric focusing. These are all ways of separating proteins based upon physical properties. Size, charge, affinity, etc. Hydrophobicity, isoelectric point. Each protein has a specific number of charged amino acids, and I'm not going to steal Doreen's lectures, but Dr. Oskin will go over this in the spring. So every protein will have some minus, some positive, some hydrophilic, and some hydrophobic, depending upon the amino acid composition that makes it up. If there's more pluses than minuses, then it's a basic protein, because it's going to be mostly positively charged. If there's more minuses, then it's going to be the other way around. So those things will affect how this thing moves in a pH gradient. It's called IEF. Each protein has a number of charged amino acids. Each protein has a unique electronic signature called isoelectric point. The isoelectric point is the point at which the protein is neutral. And you can manipulate this by changing pH. As you move the pH either up or down, you will cross the protein's isoelectric point. We should call it the PI. Separate similar proteins from each other. First dimension, two-dimensional gel is IEF. I'll show that in just a second. Two-dimensional gel electrophoresis. This is what I went over. I glimpsed this for you guys last week. IEF in the first dimension, in page, polyacrylamide gel electrophoresis in the second. 2D electrophoresis, proteins are put on a strip. Strip is exposed to electrical current. Each protein will settle down according to its charge. That's pretty understandable. In the old days, we ran these in tubes. So you would take a tube, a glass tube, very long, and put this in an electric field. Doesn't matter. We'll put a minus down here and plus up here. You apply your protein at the top. Each protein will move. So here's the protein in green. And up here, let's say it's positively charged. It will move until it reaches a point at which the net charge is 0. So as it moves down, pH is changing, and it will stop at the point where the isoelectric point is reached. As a result, you will end up with a whole series of bands. Stuff that's more positive will be down here, and stuff that's more acidic will be up here. This is called IEF, isoelectric focusing. You should run it in 2. Then you extrude this thing. This is usually gel. No SDS, no sodium potassium sulfate. This is not denatured. This is called a native gel. So it's moving to its isoelectric point. This protein stops when it reaches its PI, or isoelectric point. This is the point at which the charge is zero, either plus or negative. Everybody? And here's the 2D gel. So this is showing STSK, which is the last one. So detergent is in there. Everybody runs to the positive side. Desired protein is sub-band. This is a typical type of a gel you would see for a protein purification process. So here you're starting with some cell permeate, and your protein you can hardly see in there. This would be precipitation, maybe ion exchange, hydrophobic interaction, affinity, and you end up with your protein pure. This stuff you can now treat a little bit and shoot it into people. This you cannot, because your protein is a tiny component in all this garbage. This is called downstream purification of proteins. You have to shoot that to people and not this. If there's bands in here, something went wrong and the process has to be repeated. Lane 1 represents protein. 2 to 5 show protein purity at a sequence of steps. Obviously at step 5, the protein looks like it's about 95+% pure. And there's guidelines as to how pure it needs to be to go into humans. I showed this slide last week. IEF, isoelectric focusing, run in the tube. Charge your way, everything reaches to where it charges 0 and it stops. So you end up with proteins. These guys down here, depends if you're going to use down or use up. If this is a plus, these guys will be very negative, and the guys that will start off negative. And the guys up here will be very positive. Right? Isoelectric force will show. You then put this on top of the SDS gel. You permeate SDS into there to denature everybody, and then you run it in the second dimension. You get a picture that looks like this. This looks horrible. The 2D gels, running 2D gels is apart, and there are people that can do this and routinely produce, say, 1,000 or more spots. And these represent proteins that are present in a cell lysate or secreted in something. Every spot is a unique protein. Does everybody understand? So stuff separating this way will be positive and negative based on IEF, and stuff separating this way is based on molecular weight. Now, by an incredible technique, which comes in the next few slides, called mass spectrometry, this technique is awesome. You can cut a spot out, so you can take a little razor blade and cut that out of the gel, mash it up, and determine the amino acid composition and the sequence. And this technology all came available about 30 years ago. Mass spec on tiny scale. You can determine what that spot is. Usually, often people run 2D gels on cell lysates or cells that have been treated differently. So you can take bone cells and treat them to either make bone or degrade it, and then look at which spots differ, and then determine what they are. This is a very powerful technique called the 2D gel. Analytical methods, high HPLC, high performance liquid prototype, we'll talk more about later. High pressure blasts everything through. The beauty of high pressure is columns and pumps don't go that fast. With HPLC, you can have separations take minutes instead of hours. Less protein, these are for analytical labs. We just want to say, for example, how pure is my protein? Okay, that was a John Bennett pen. Received the Nobel Prize, jet propulsion, awarded Nobel Prize 2002. This guy developed ESI, electrospray ionization. I know it doesn't mean anything to you guys. This is called a quadrupole mass spec. These These usually cost about $1 million. And most labs have, we had like 4 or 5 of these. And whole teams of guys that work on it. Quadruple mass spec. Using this machine, you can cut that little spot out, and the next day the guys will say, okay, here's the amino acid sequence, and here's the composition. It'll tell you the exact composition of the amino acid making up that little spot on your check. Very powerful. And you need nanogram quantities. It's incredibly sensitive. Brilliant technique. ESI. Mass spec enables protein detection, quantitation, and identification, composition, and sequence. So what the exact sequence is from the N-terminus to the C-terminus, you can determine with this method. It's just a total breakthrough. And when it occurred, obviously, all labs and companies immediately purchased this. TOF, we spend a lot of time, this is time of flight, and I'm not able to do this. It's called time of flight. Oh, I guess maybe not. Tandem mass spec. Determining AA sequence of given protein yields primary MO versus E secondary. Ion fragments are separated. This is all called time of flight. Where's this going? Okay, what should you be learning out of this? What you should be learning out of this is what I just told you. There's a little bit too much detail. What this does, it allows you for any protein, must be a protein, and you can have very small amounts. You can determine AA composition, and you can determine AA sequence. That's what you should do, using mass spec. Composition will say you have 36 alanines, 42 glycines, 3 methionines, etc. Give you the breakdown of the big 20. This says position number 1 is a MET, position number 2, etc., etc. Okay? Everybody got it? This is called mass spec. Mass spectrometry. Non-magnetic electrospectrum. This is called time of flight. I don't want to explain it. It's massive magnets. It's $1 million instrument, so obviously there's a lot of technology. Analytical methods used for protein separation. Mass spec sensor method identifies small differences. Used as the outflow of HPLC. So HPLC is just high performance or high pressure liquid chromatography, which allows you to separate stuff very fast. LC is a column. In HPLC, they're steel jacketed and they withstand high pressures because you're blasting stuff through with a powerful pump. This allows you to do separation in minutes instead of hours. Why is that so important? Time is money. If you can get your result in 1 hour instead of a day, that's worth a lot. Mass spec does 3 things. Suspend sample of the charged data space, separate molecules based on mass charge ratio. This is part of this time of flight. Detect separated ions, read out, produce interface identity and size. And with electron spray, you can determine, see each amino acid in non-copy. Analytical methods, mass spec, protein sequencing. This technique, first protein is ingested in little pieces, chopped up, and then you determine the sequence. Just like massively parallel sequencing, next gen, very simple. SDS page, detergent is added, and this neutralizes all the positive charge. The protein is negative, just like DNA, that flows to the end of the gel that has the positive electrode in it. Sulfate charge is evenly distributed. It's treated with SDS samples located at the page, forms a single band in some location based on molecular size and mass, just like a agarose gel that you guys are using. Kumassi stains proteins blue. We use a ladder. Remember, we use the DNA ladder in other labs. Purification process color band is increasingly intense, showing protein velocity. Verification. Proteins are transferred from aspartate to nitrocellulose. This process is called blotting. Let's see if anybody's still doing it. Blotting. And remember, why is it a good thing to blot proteins? Pardon?
It separates them out.
I wouldn't put it quite like that. So you've already separated the proteins out on the gel, right? You now sandwich the gel with the nitrocellulose membrane. You drive all the proteins onto the membrane, so now it's on this nitrocellulose gel. Now it's on this membrane. Why? Why is that important? Who cares? I mean, the sequence is the same. It's not a separation, right? Because the separation is already done in the gel. You turned a three-dimensional blob into a two-dimensional surface, right? Everybody remember that? So if you look at a gel, perfect, thank you. You look at a gel, it has thickness. And your proteins are all embedded in there. When you transfer it onto a blot, if you look at a blot, now on the edge, so here's a blot. The proteins are all forming little mounds on here. Secondary structure is accessible. This means epitopes. So for example, you can do a Western blot. You cannot do it on a gel because the antibody will not get through the matrix. Okay? So that's why. It was a good guess, but no separation. All you're doing is transferring into an accessible form. I know you were going to answer it, but I wanted to get something. What else is on here? Sites on membrane are blocked, for example, using non-fat dry milk. Primary antibody is added. Everybody get this right on the quiz, I hope. Primary is something that you make or engineer, and it's detecting an antigen of interest. In the case of chicken gamma globulin, that was the protein of interest, and the primary antibody detected it. If there was any antibodies present, binds. Detect antibodies of the bound protein. You chuck in a secondary. What does the secondary have? Say it?
Enzyme.
Enzyme. What does the enzyme do?
It attaches to a substrate that makes it visible.
Some S substrate is converted into a P, and the P is color. And you see color. I think in the lab we did it failed the last. It turned blue, and we figured it out, by the way. It was not your fault. It turned out it was my fault. Detect antibodies in the secondary. The secondary is conjugated and has some sort of tag, usually an enzyme, such as horseradish peroxidase, HRP. Secondary is washed away, substrate is added, you end up seeing color, and everything's beautiful, and this is called a Western blot. So here's a page gel, and here you see your band lighting up on a blot. ELISA, I also went over this in the last lecture. 2 antibodies, procedure takes place in plates. Antibody 1 captures, and antibody 2 recognizes this, has some sort of conjugate and an enzyme so that you can see color. So how do you preserve proteins? I mean, once you've made kilos of it, you want to ship it to customers around the world. How do you do this? Once protein is isolated, collected, and purified, it must be saved to preserve its activity. Ways to preserve. Lyophilization is probably the most important. Freeze drying is removing all the water. Why don't I stop on this slide, and we'll reconvene at 11:05. Sorry, 11:05. Communication difficulties, lab methods may work on the small, may not always work large. Changes in the purification system can invalidate small-scale clinical studies. Why is it often necessary to seek FDA approval again for an approved bioengineered protein when the protein purification process is scaled up? For exactly this reason. Often it changes. So if you go to the FDA with a small scale and you say, okay, now instead of running a little benchtop bioreactor in a column this big, I want to run a column that you can hug that's 10 feet wide, and I want to run a 3 or 4 story high fermenter. They'll say, how are you sure that it's going to purify exactly the same way? Many things don't scale well. Some do, some don't. Okay, so that can be a problem. If you change any aspect of the manufacturing process, you swap a column, or you grow it differently, or you change something, you have to inform the Food and Drug Administration. And they may say, well, no, I think that's significant. You can't just do that. You're going to have this whole thing reassessed. You have to seek approval a second time. Post-purification analysis. So how do you know that your protein is pure and that it's what you think it is? Determine primary amino acid sequence. So sequencing. Mass spec, which I just described. Peptide masses are identified by unique signatures. Possible to identify many unique proteins in a short period of time. Minute quantity sufficient. This is a summary of the John Bennett Fenn MS slides, mass spec slides that I just did. Now, CMS, which is liquid chromatography, followed by mass spec like quadruple, the big $1 billion systems I described, can be conducted on spots, X size out of page shells in which protein has been eluded. That's what I just said. We cut a spot out of a 2D gel, send it into the lab, and they will do the two different phases I described. Determine the molecular weight, that will give them the amino acid composition, and they can determine sequence. Protein spot proteolysis, trypsin, etc. Frequently does generate what's called a tryptic map. I wanted to say one more thing really quickly here, which is something called reverse translation. I just wanted to mention it. This is all done in silico. So reverse translation. This is all done in silico. What you do is you take an amino acid sequence, so AA1, AA2, AA3, etc. And from it, you predict mRNA sequence and DNA sequence. It's called reverse translation. This is not done by any organism on Earth other than computers. Okay? Now, what good is it? Because from that spot that you cut out of a gel, you can determine the gene. You can say, I know which one of the 20,000 human genes codes for that spot. Now, question. How do you reverse translate? So what's the problem going to be? So you know the amino acid sequence? Can anyone here predict, given the genetic code, what the mRNA and the DNA sequence will be? Yes or no? So if I gave you a sequence of amino acids, say 3 or 4 of them, and I gave you remember the gene code table, which takes each triplet codon and assigns it to amino acid, would you be able to predict mRNA sequence? Yes or no?
Yes.
No.
Lauren, can you break the
Could
tie?
narrow it down so we can actually look at those specific.
Correct. The answer is not yes. Why? Because many codons code for one amino acid. If you guys will remember, leucine has 6. There are 6 codons. Now, because of the wobble position, which is the third, usually any of the 4 will code. You can know pretty well what it is, but not perfectly. But given something called codon usage by an organism, you can get a good idea. And you can try a bunch of different sequences. All you need is about 5 or 6 amino acids. 5 times 3 is 15, 6 is 18, that's going to be unique. Blast the database and see if you can fish the gene. It's generally pretty reliable. If the answer is no, and the answer is you get partial, but not perfect. It's a good try. There are too many codons for one AA. You'll never know. Good guess. X-ray. This is the structures I show you where you're seeing helices and sheets and stuff. That's all done by X-ray crystallography. Used to determine complex tertiary and quaternary structure. It tells you essentially where every atom is located. Question?
Thanks.
Any questions?
Yes, I'm sorry for interrupting you. I just wanted to ask if what that word was underneath reverse translation in
silico.
In silico, okay.
Right, so this is something you
Okay.
do with a computer. You cannot guess.
Okay.
You can guess, but
Okay.
generally you'll have, 5 or 6 guesses that look like they'll be pretty good, and you can just have the oligos made or blast it and see what you fish.
Thank you.
Sorry, in silicon, scribbling.
Thank you.
You need crystals. Not all proteins crystallize. Transmembrane proteins like receptors are bad because of detergents and the hydrophobicity of the bilayer. They're often very, very hard to make them crystallize. Exposed to X-rays. X-rays are tiny, smaller than atomic distances. So the resolution of X-rays can often be angstrom, 1 angstrom or less. Pure protein contains specific shadow patterns. Same thing with DNA. Remember Rosalind Franklin, who got the X-ray structure of DNA, and Watson and Crick used it to predict the distances, and that allowed them to build their model with the stack basis. Computer analysis of patterns generates ribbon diagrams, and then you know exactly where all of the amino acids are and what they're all doing. FDA requires protein crystal for approval. Verified protein is non-denatured. Folded correct structure has been achieved. Proteomics, new scientific discipline. It's not new anymore. This has been out for at least 20, 30 years. Dedicated to understanding complex relationship between disease and protein expression. Proteomes. Proteomes means the total proteins produced by a cell or an organ type or a living system or organism, compared between healthy and diseased states. So for example, you can take cancer cells versus normal cells from the same tissue. So for example, ovarian cancer, very common in women, or cervical. So you can look at normal cells versus disease and say what proteins are different. For example, 2D block, this comes up on the exam, I've written this every year. Run a 2D gel, compare it to biopsy from normal ovary or cervix and an abnormal. See what spots are different. John Bennett Fenn, MS. You can determine and predict the gene. Reverse translation. Got it? Discover protein markers using diagnostic methods. Proteomes frequently assessed by 2D electrophoresis. The 2D system I just went over. IEF, SGS page.
For the X-ray crystallography, you said that not all proteins produce pure crystals. Does that also mean that you're not making biologics for the protein that is in crystals?
Correct. Or that's correct. You usually need a soluble portion. So whole receptors are never biologics. But soluble portions that do crystallize can be. So you often have to engineer the protein so it has the properties that you want so that it is crystallizable. 2D gel followed by mass spec, verified proteins. Use amino acid sequence to characterize protein. Protein microarray. This is the third type of microarray. I described DNA and RNA just before at the first part of this lecture, based on your exam. Protein microarrays exist. The proteins are mobilized on a surface. Coded reagent indicates color. Functional protein microarrays recently implied discovery of all kinds of interactions: protein lipid, protein DNA, protein drug, etc. Microarrays. This is showing some capture antibody, which is in this represents one of those little dots in a microarray with, say, 100,000, and you have some antibody there. So same process. You do photolithography, and you put antibodies in certain locations that detect things, then you can see color produced. So protein microarrays uniquely bind single protein, release some signal. Chromogenic means something's changing color or it's fluorescing. Enzymes such as is an example of this. Microarrays, amino acids, protein-protein interactions, protein-lipid, protein DNA, small molecule, peptides, kinases, immune responses, etc. All those. Finally, this chapter the rest. Okay, this now I'm going to finish. This is fairly easy. This stuff's uploaded. I loaded it up last Friday. I need to do this because this is your pre-lab right now. This is what you're doing in lab today. Any questions so far on this whirlwind chapter that I did? Anybody have any questions? Everything good? Cell culture. Cell culture means literally culturing cells. What you guys do today in lab is we're going to start off with a vial about so big. And this is frozen. And this has chosen. Now, the students from last year froze these down, just like you guys are going to be freezing these down in another 2-3 weeks when you're done. Your job is exactly what we do with DEX, with hybridoma and biomanufacturing. We stuck this in here so that everybody would have an experience with it, so that biomanufacturing is not a total disaster like it was a number of years ago before we was. Everybody's cells were contaminated. I never had enough to go into the bioreactors that I was doing before. So you guys are going to thaw this thing up, thaw, and then you're going to wash the cells. These are CHOs, tiny stands for ovary cells, and then you're going to plate. And you're going to plate them in T25s, the same one that we did for the aseptic lab. You guys will all look at your plates. I don't think anybody got contamination. Well, anyway, last week they were fine. So I don't think anyone's been contaminated. Remember, you put media in, and our goal is to see if tons of stuff grows. If something grows, then it means that your technique needs improvement, or you coughed or something into the pudding. Okay, so what we're going to do today is thaw this up, wash the cells, and start to grow them on a T25. T25 is that little flask that you guys use with the cap. You put it on its side, remember? So what is all this involved? Because we're doing this today. Cell culture during upstream processing divided into two phases. Laboratory scale, this is always the first part. So everybody, even Amgen, starts with a little vial of frozen cells. Again, there's an officer of the law, and he checks you in. You take your vial, sign a book, and he escorts you out of the vault, which has thick glass. You can see it. Occasionally, you see guys walking in there, getting stuff. Exact same thing, except we don't have the vault. We just have the freezer. Dividing line between the two is some arbitrary and laboratory cell cultures can be used as a stir flask from 1 to 10 liters in capacity. So usually lab scale is something that gives you a few liters. This is giving on the order of milligrams of purified protein in there. Okay, that's called lab scale. Large scale will require apparatus from 100 to, this is a mistake, this can go up to 20,000 liters and more. The big bioreactors at Amgen are 20,000 liters. Okay, and you'll see them, and they all started with one vial about this size. And they end up in something about the size of this room. Full-scale industrial production, 20,000. Stir culture or spinner culture are synonymous. Spinner flasks you guys will use. I don't think maybe this semester, but next semester. Fermenter and bioreactor are not synonymous. Synonymous, and this felt considerable overlap. Fermenter, used for use in microbial systems, yeast or bacteria. Usually grown in a fermenter. Bioreactors usually refers to vessels for animal cell culture, such as CHOs or DEX, either monolayers or suspension. This is usually microbes, and this is usually animal cells. Method employing increased scale depends on whether the cells proliferate in retention, suspension, or anchor. This is called detachment dependent. That means the cell has to sit on a surface and usually spread out and form some kind of morphology. More on this next semester. Must be anchored. Methods for growth of cells suspension are generally simpler because the cells are bobbing around and free swimming in solution. CHOs will stick. Note, anchorage-dependent cells, monolayers growth, must continually be supplied with increased surface area because they're growing on a substrate. What is an example of an attachment-dependent cell? Any cell that you can think of in the human body, for example, that grows on extracellular matrix or something that requires a wall or a board. An example is epithelium. Epithelial cells are usually linings of organs like lungs or GI tract. So those cells are attachment dependent. They need to be stuck on something. Okay, it's a single layer of cells, much more complicated than this is. Okay? Everybody all right? We got through last chapter. This is, easy. This is kiwi. Nature of substrate where cells grow. Solid, semi-solid, liquid. Way more detail in the next one. Just accept it. You can grow stuff on plastic, glass, some sort of gel. This is called three-dimensional cell culture. Degree of contact with other cells, physicochemical and physiological constitution of media, constitution of gas phase. Obviously, you need oxygen and you need CO2. CO2, you need what? Remember, CO2 incubator. All you guys' cells are in there. Why do you need CO2? Thank you, Dave. It's called the CO2. Remember the bicarbonate? It's the bicarb buffer system. CO2. Incubation periods, how long you leave it in. Appropriate environment is fundamental to expression of specialized cell function. Way more detail in the next measure. Cell adhesion, this is called anchorage dependent. Epithelial cells, fibroblasts, stuff like that, form layers. Mediated by specific cell receptors for molecules, extracellular matrix. No one will remember this, but when I showed you guys the video a long time ago, it showed adhesion molecules. Okay, and adhesion molecules are things that are binding and then allowing cell surface contact. I can't. It's originally found cells were attached and spread on glass that had a cell negative charge. Matrix adheres to charged substrate and cells bind the matrix by receptors. Substrate has been conditioned by previous cell growth and often provide a better surface for attachment. Glass is still used a lot today, but mostly plastic. So most growth today is disposable, right? Because it's cheaper. You don't want people washing and sterilizing stuff. Lots of things are grown on plastic. The T25 plastic you guys use are aseptic sterile, and they're just, as soon as you guys are done, you throw them out. Suspension, primarily increase in volume because you don't need a surface. The guys are bobbing in solution. Agitation of media is necessary, depending on surface area volume. Sparging with CO2 and air, oxygen, buffer, bicarbonate buffers. Stirring of such cultures is done with magnetic, encased in glass or other non-metallic substance. There's the pendulum type of, I think we go over this next. And all kinds of the bioreactors have a motor-driven impeller, propeller that goes around really slowly and keeps everybody suspended. Stirring speeds usually somewhere around 60 revolutions per minute, 1 rotation per second. If you want to keep cells suspended, but you don't want to start breaking them. Mammalian cells are very delicate. They're not like plant, bacteria, or yeast. Those guys all have cell walls. Antifoam is often used, so if your cells foamizes up, and you lose sterility. Absence of serum necessary to increase viscosity of media with CMC or various types of agents. So it makes it a little more viscous and everything's happening. Typical pictures, rather low quality. This shows bioreactor and all these cables coming out of it are giving you pH, dissolved oxygen, cell number, etc. This is another stirred tank. These are other types of stirred tank vessels. All of them used for scaling. Wave bag. Wave bags are awesome and just exactly like it sounds. Huge plastic, these things can be scaled to their, about 6 feet across. And they're on a gigantic table that goes like this. And as it goes, there's a wave that goes, wave goes back and forth, see the media in it. This is all sterilized by gamma radiation called wave bags. Beauty of wave bags, infinitely scalable. Amgen just built, I doubt we'll get to see it, but Amgen just built a whole new building. It has a small footprint. It's supposed to be high efficiency. It's all based on disposable wave bag technology. So instead of the giant steel tanks, they're expensive, they have to be sterilized in place. It's called SIP, steam in place. These you pitch, so of course enormous amount of garbage. But despite that, you can make this stuff fast, cheap, smaller footprint. So they have a whole building filled with wave bags. And they're huge. They can be, 6, 10, 12 feet across. You have hundreds of liters in there. Slops back and forth. And that's called a wave bag. This is called a liver bioreactor, which came out some time ago. This is a human liver or an animal liver showing the liver right is this hyper-perfused system where cells are all present in a matrix. And this is basically mimicked in plastic. And allows you to grow huge numbers of cells, essentially in 3 dimensions. The cells are contacting each other, and they're growing in little orifices within this matrix. It's called a liver bioreactor. Liver bioreactor designed for growing millions of cells in a 3-dimensional array. We'll talk about 3D technologies more in the spring. I'm not going to touch it. Massive tissue needs sensitivity requirement, many types of analysis. Extract again experiment samples of media. Primary means for gathering experimental details, evaluation of therapeutics, etc. Done in these types of scale up bioreactor capacity for millions of cells. Cells residing in scaffold with hundreds of stacks and channels. This is still the liver bioreactor, 300 microns in size, 3 components, scaffold, 3 component functions, optical window allows you to assess what's going on in there. Cells going in suspension or each year anchored cells have greater potential for developing polarity. Polarity means the side of the cell that's stuck to the substrate is not the same as the other side. And if you guys will remember brush border, for example, in intestine or airways, they're highly polarized. So there's an attachment anchorage part, and there's usually a brush border, which is changing gases in the lungs or food and digestive or post-translational modification itself. Anchorage-dependent cells are monolayer culture necessary to increase surface area, so you've got to get more plastic. This process is called scale-up. This is a triple flask, which obviously has 3 layers. You guys had a single layer on a T25. T25s are small and cheap. This is getting more expensive. Notice, this is one of those gas exchange caps. So there's fritz here, and there's mesh that prevents bacteria from entering. These, remember how you guys backed off a corner, half turn, made sure it was loose to enable? Here, gas can flow right through this frit. That's called a gas exchange. Nunk cell factories. This is we had these in the companies. In the company I worked at, a dozen of these. These are big. They're, again, these can be many feet across, stacks this high, and they're often gently agitated, and stuff grows. You can grow billions of cells here. So this is all part of scale-up. Remember, take one little frozen vial, and you end up here. So this is packed with cells, and one port has media going in, you go gas exchange, and then media and hopefully product goes out. Everyone got it? Cell factories, cultured monolayers, the advantage they do not alter geometry or nature of substrate from that of conventional dish or flask. So it's plastic layers just like before. Roller bottles is exactly what they say. This is another common technique. We use these for medium scale, but usually cell factories if you needed a ton of recombinant protein. Cells are seated into a bottle, and the thing turns slowly, like very slowly, there's about this fast turn. non-adherent magnum media, and adherent, they form a monolayer all around the inside of the bottle. I'll show you a picture. Roller bottle, increased usable amount of surface area, constant gentle agitation, increased ratio of surface area to volume, gas exchange is favorable, etc. And there's a film of water that covers the cell at all times. So this shows a typical roller bottle, which is this cylinder. Let me show you. Typical roller bottle, and this is the machine they roll in. This continually turns them very slowly. It's about a revolution a minute. So it turns, there's media in the bottom, and attachment cells will go all around the inside. And there'll be a film of water around the inside, massive gas exchange. These would need to have the cap loosened. And then you can harvest by sweeping the cells off the entire inside of this bottle. So it has a huge surface area. This is a typical roller bottle machine. It has to be dead centered because you don't want the liquid to be pitched up or down in the bottle. You want the bottle absolutely level with gravity. So you're forming a constant liquid layer. Everybody got it? That's called a roller bottle. We do not do that here. You need you can't do it. So how do you count cells? We do this next week. I'm not going to present this week. We do it next week, and I'll spend a good 45 minutes at the end of lecture explaining how to count cells. Cell counting often accompanied by viability staining. This is called trypan blue, which we'll use next week, and I'll explain about it more. Penetrates membranes of non-viable cells, but viable cells remain white against the blue background. Percent viability is calculated, percent viability number of viable cells divided by total number of cells times 100. This is called a Neubauer hemocytometer. I will explain this next week. You guys will use it. But next week you will count cells using this system. I will explain it. Microcarriers, very popular. We had this when I was at Biogen. I worked at Biogen for a couple of years. Microcarriers, these are often made of digested collagen, like from cow hides. You can digest collagen and break it up and form these microcarrier beads. And these are packed with cells. So attachment dependent, but the microcarrier is in liquid. We had a huge bioreactor that was about 3 or 4 feet in diameter and about 20 feet tall. It was like a giant lung. It was 37 degrees. You could go up to it, and the huge pump, boom, and you'd see that it would pump fresh media and liquid in from the bottom and keep the cells suspended, and out the top would flow recombinant protein. So this is called a microcarrier. So it's used for anchorage dependent cells that need attachment. But the whole bead is suspended in liquid. So it gives you a gigantic surface area. Sound reasonable? Very, very, very effective. It's called a microcarrier. How do you count? How do you count these things? What do you do? Easy. A modified version, the method using crystal violet is used. You cannot use a cytometer because the beads are embedded inside of this matrix. So each bead, cells are crawling all through it. It's packed with cells. You add crystal violet and it doesn't mention citrate. Usually add citrate in crystal violet. It busts the cells open and you count nuclei. The nuclei flow out of the bead. So you can count nuclei. Cell suspension, like here, citric acid, crystal violet. Cells bust, and you look at purple, count nuclei. And we did this routinely when I was 5. Many cells from culture become binucleate. Care must be taken. Growth slows or becomes arrested. Cells become binucleate. And enough cell concentration sometimes actually overestimates numbers of cells. Errors in counting result from all kinds of stuff. About 10% error, variability in sampling, dilution, preparation, filling the chamber, counting cells. So there's a user error. You guys will be doing this. You're staring through the microscope at that grid, and you've got a little clicker in your hand, and you count the cells in the go. So you guys will all be doing it. Click, 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 click as you count cells. Well, if you miss one, your count is off. So that's hemocytometer. You'll do this next week. And that's references, and we are done. Whoa, record time. Any questions about cell culture? And I'll go over what we're doing in lab at 12:15. Remember, lab quiz. We have a lab quiz, 10 or 15 minutes. It's on all the labs up to now, not including cells. Okay? Good luck. See you in 35 minutes. Any questions?
Matt,